Fundamentals of Immunology within Immunohematology. So in introduction, we have immune mechanisms. So we have a cellular defense mechanism, and this involves immune cells such as macrophages, T cells, and dendritic cells. These cells play a critical role in identifying and destroying pathogens. The macrophages engulf and digest microbes and present antigens to T cells. T cells recognize infected cells and help coordinate the immune response. And dendritic cells capture antigens and present them to T cells, initiating that immune response. Then we have the humoral mechanism, which is primarily mediated by B cells. The B cells produce specific antibodies that target antigens on pathogens. The complement system is a group of proteins that bind to immunoglobulin molecules or antibodies. Complements enhance the ability of antibodies in phagocytic cells to clear microbes and damaged cells. Complement proteins have specific receptor sites on immunoglobulins, which trigger a cascade resulting in the destruction of the pathogen. So detection of alloantibodies or autoantibodies in routine blood bank testing procedures is of the utmost importance in providing compatible blood to patients. This detection is dependent upon several factors, which include binding forces between antigens and antibodies, properties of the antibody itself, and individual host characteristics. Antigen-antibody reactions are influenced by a number of factors, including distance, antigen-antibody ratio, pH, temperature, and immunoglobulin type. So alloantibodies, these are target antigens on red blood cells from other individuals. They are produced after exposure to a foreign red blood cell antigen, such as during pregnancy or a transfusion. Alloantibodies can be clinically significant and can cause hemolytic transfusion reactions or harm a fetus or newborn. And then we have autoantibodies. These target antigens on a person's own red blood cell. They can be caused by an autoimmune process, which can primarily or secondary to other conditions like infections or lymphomas. Autoantibodies are usually not significant, but they can cause problems with blood bank compatibility testing. So both alloantibodies and autoantibodies are associated with red blood cell transfusion. So before each transfusion, antibodies must be identified in the recipient's serum to ensure compatible blood is provided and given to the patient. So as mentioned earlier, we have different types of antibodies. We have autoantibodies, which are directed against the self-antigens. These antibodies mistakenly target and react with the person's own tissues or organs, and it can contribute to autoimmune diseases. Alloantibodies are directed against non-self antigens. These antibodies react with antigens from other individuals of the same species, commonly seen in transplant rejections or blood transfusion reactions. An antigen, it is a molecule found on the surface of a foreign cell or damaged internal cells. Antigens are recognized by the immune system as a target for immune response. All right, so characteristics of immunoglobulins. We have heavy chains. The antibodies or immunoglobulins are composed of heavy and light chains. The heavy chains determine the class of the antibody. So the types of heavy chains, we have the gamma, which is the immunoglobulin G or IgG. This is the most abundant antibody in serum, and it provides long-term immunity after infection or a vaccination. Then we have the alpha chain or the immunoglobulin A or IgA. This is found mainly in mucosal areas like your respiratory or gastrointestinal tract. These play a crucial role in mucosal immunity. And we have the mu or the IgM immunoglobulin M. It's the first antibody produced in an immune response. It exists primarily as a pentamer 4 in the serum. Then we have the delta chain or immunoglobulin D or IgD. It functions mainly as a receptor on B cells. Role in immune activation is not well understood quite yet. And then we have the epsilon, which is immunoglobulin E or IgE, and it is involved in allergic reactions and protects against parasitic infections. So the most significant immunoglobulins in blood banking is IgG, IgM, and IgA. We have certain temperatures that these will react at. So antibodies of the IgG class will typically react at your body temperature, which is what we don't want to happen. Naturally occurring antibodies, so most naturally occurring antibodies are going to belong to the IgM class. Commonly encountered IgM and IgG antibodies that we will talk about later on in this course in blood banking are your Lewis P and MNS antibodies. It's a video for you to watch on the complement system. So characteristics of antigens. So antigen characteristics that can influence an immune response is due to size. So the larger molecules are generally more immunogenic because they can be better recognized by the immune system. Their complexity. So complex molecules with multiple different components tend to stimulate a stronger immune response. The conformation. So the three-dimensional shape of the antigen affects how well it fits into antibody or receptor sites. Charge, which is the electrical charge, it can influence how antigens interact with immune cells and antibodies. Accessibility, so antigens exposed on the surface of cells or pathogens are more likely to trigger an immune response. Solubility, so soluble antigens are often more easily processed and presented by immune cells. Digestibility, so antigens that can be broken down and presented by antigen-presenting cells tend to stimulate immunity more effectively. And then we have the chemical composition. The presence of proteins, carbohydrates, or even lipids can help influence how the immune system recognizes and reacts to that antigen. Just an additional note, so blood group antigens vary in their immunogenicity, and that means some are more likely to provoke antibody formation and immune responses than others. So this is important in blood transfusion and transplantation because we don't want to have any adverse reactions for our patients. So characteristics of blood group antibodies, we have polyclonal antibodies. These are produced by different clones of B cells. They recognize multiple epitopes on the same antigen, and they're usually found in natural immune responses, and they are used in diagnostic testing because they detect a broad range of antigen sites. And then we have monoclonal antibodies. It's produced by a single clone of a B cell or a hybridoma cell. It is recognized by a single specific epitope on an antigen. They are highly specific and consistent, and they are widely used in blood typing and diagnostic assays. And then we have naturally occurring antibodies. These are formed without prior exposure to transfusion, injection, or pregnancy. They typically belong to the IgM class. They are active at room temperature or lower. They can activate complement, which leads to cell lysis. They may be hemolytic at body temperature, which is at 37 degrees Celsius, and that can cause clinical significance. Some common naturally occurring blood group antibodies is we have the ABO or ABH system, so anti-A, anti-B, the H system, the I system, Lewis system, MN system, and P system. Just this little side note, so these systems are important in blood compatibility and transfusion medicine, and we will discuss all of these in detail later. Immune antibodies, so their origin is produced as a result of exposure to a foreign antigen through transfusion or pregnancy. The class is generally, it's an IgG antibody, and they usually will react at body temp. For detection, it's going to require the use of an anti-human globulin or an AHG test for identification because they do not usually agglutinate red cells directly. Common blood group systems that are involved in this are your RH system, so your anti-D, anti-C, and anti-E's, the Kell system, the Duffy system, KID system, and S system. We have naturally occurring antibodies or ABO antibodies as well. These are called isoagglutinins. These are antibodies naturally present in the plasma against A or B blood group antigens that an individual lacks. So these antibodies develop spontaneously in individuals within the first few months of life without any prior exposure to red blood cells or transfusion. So these are significant in blood bank because, well, ABO antibodies are highly significant because they can cause immediate hemolytic transfusion reactions if incompatible blood is transfused. They are also responsible for the major ABO incompatibility in blood transfusion in organ transplantation. For unexpected antibodies, these are antibodies other than the naturally occurring IBO isoglutinins, or ABO isoglutinins, may arise due to sensitization from pregnancy, transfusion, or exposure to environmental antigens. And these are often clinically significant and can require a thorough detection to find them. So some detection techniques is we have an antibody screening. This tests patient's serum against a panel of reagent red cells to try to identify the unexpected antibodies. We will also do cross-matching, and this is testing donor red cells with the recipient serum before transfusion, and this helps to ensure compatibility. We have direct and indirect antiglobulin test, or a DAT and an IAT. These may be used depending on which antibody we are suspecting. And we'll talk about those tests later in the course as well. So we have importance in pre-transfusion testing. So this is to detect ABO antibodies. Just, basically helps to ensure a safe blood transfusion, helps to prevent hemolytic transfusion reactions, and helps determine the correct blood group and compatibility. Alloantibodies are produced after exposure to genetically different or non-self antigens, such as different red blood cell antigens after a transfusion. So transfusion or transfused components may elicit the formation of alloantibodies against the antigens, which could be on your red blood cell, white blood cell, or platelets, and not present in the recipient. Autoantibodies, on the other hand, are produced in response to self-antigens. They can cause reactions in the recipient if they have a specificity that is common to the transfused blood. Some autoantibodies do not have a detectable specificity, so autoantibodies can react at different temperatures as well, and both cold and warm autoantibodies may be present. So patients with autoantibodies frequently will have autoimmune diseases. Autoantibodies can be removed from red blood cells by special absorption and elution techniques, and then tested against reagent red blood cells. So therefore, in order to transfuse blood safely, we have to identify the antibody and determine it and record it. That way the patient does not get that type of blood. So traditional laboratory methods for testing is we have hemagglutination, so methods for analysis of blood group antigen antibody responses, and typing for ABO-RH and other blood group antigens is accomplished by the red blood cell agglutination reaction. We have precipitation as well, which is a lattice type structure, which is composed of multiple antigen antibody bridges between red blood cell antigens and antibodies is formed. A network of these bridges forms, and visible agglutination is present during this stage. The development of an insoluble antigen-antibody complex resulting from the mixing of equivalent amounts of soluble antigen and antibody. Agglutination inhibition is a method in which a positive reaction is the opposite of what is normally observed in agglutination. Hemolysis represents a strong positive result and indicates that an antigen-antibody reaction has occurred in which complement has been fixed and red blood cell lysis occurs. So the Lewis blood group antibodies, anti-Lewis A and anti-Lewis B, may be regarded as clinically significant if hemolysis occurs in their vitro testing reactions. And like I said, we'll talk about those later on in this course. And then we have ELISA testing techniques, and these are immunologic methods, which are based on detection and quantification of antigen or antibody by the use of enzyme or fluorescent labels. And either the antigen or the antibody can be labeled in these tests. So centrifugation is an effective way to enhance agglutination reactions because it decreases reaction time by increasing the gravitational forces on the reactants and bringing reactants closer together. So high-speed centrifugation is one of the most efficient methods that we use in blood bank. The zeta potential, so under the right centrifugation conditions, sensitized red blood cells overcome their natural repulsive effect, which is the zeta potential, on each other and agglutinate more efficiently. Having red blood cells in closer physical proximity allows for an increase in antigen-antibody lattice formation, which will result in enhanced agglutination. Then we move on to antigen and antibody. They have optimal concentrations. So in ideal reactive conditions, an equivalent amount of antigen and antibody will bind. Any deviation from this can decrease the efficiency of the reaction and a loss of the zone of equivalence between antigen and antibody ratio, which is actually necessary for agglutination reactions to occur. So if an excess of unbound immunoglobulin leads to a pro-zone effect, and a surplus of antigen can lead to a post-zone effect. Some more factors that can influence agglutination reactions is pH, temperature as well, different techniques that we can do for IgG and IgM. Like I said, we'll talk about these later on, so this is just kind of a list for now. And same with these, we will talk about these as well. And then we have monoclonal versus polyclonal reagents. So traditionally, polyclonal anti-sera reagents have been produced by immunizing donors with small amounts of red blood cells that were positive for an antigen that they lack, and then collecting the serum and isolating antibodies against that antigen. So AHG, or anti-human globulin reagents, were originally made by injecting animals, which were usually rabbits, with human globulin components and then collecting the anti-human antibodies. One antigen can have a number of different epitopes, and polyclonal reagents are directed against multiple epitopes found on the original antigen, which was used to stimulate antibody production. Monoclonal reagents are directed against specific epitopes. They are made by hybridoma technology with spleen lymphocytes from immunized mice that are fused with rapidly proliferating myeloma cells. The spleen lymphocytes have single epitope specificity, while the myeloma cells make a vast amount of antibody. After extensive screening and testing, these are efficient hybrid cells are selected and cultured to produce lines of immortal cell clones that make a large amount of one type of antibody that reacts with one specific epitope. Monoclonal reagents do not use human donors and therefore do not use a human source for reagent purposes. Diseases important in blood bank serologic testing, so we will talk about these later as well. And same with this.